Welcome, learners. I'm Dr. Seema Desai Fernandez, Assistant Professor, Department of Botany, St. Xavier's College, Mapsa, Goa. This program is for TYBSc Botany student, Semester 5. Paper code is BOC 106. Title of the unit, Unit 3 Cell Division, Module name Meiosis, Module number 28. Outline, Meiosis cell division, types of phases, significance of meiosis. Learning outcomes describes the meiosis cell division, explains the types of phases, gives the significance of meiosis. What is meiosis? Meiosis is a type of cell division where a single cell divides twice to form four daughter cells which are haploid, each with half the number of chromosomes as the parent cell. It is found in almost all eukaryotes, animals, plants, fungi, and protists. Original cell is diploid, that is, it has two copies of chromosomes. It is necessary for sexual reproduction in eukaryotes and produce the sex cells or gametes, that is, eggs in females and sperms in males, in animals and spores in plants. Meiosis process involves two nuclear divisions. Meiosis 1, the first time the cell divides, and meiosis 2, the second time it divides. During meiosis, DNA replicates once, but the nucleus divides twice. Meiosis 1 is sometimes referred to as reduction, and meiosis 2 is referred to as division. It is also known as reductional division because it reduces the number of the chromosomes to half the parent cell. This is the figure which explains the meiosis cell cycle. The cell cycle starts with the interphase, that is G1, S phase, and then it goes to G2 phase. Here, the cell prepares itself to go into the next division, that is the meiosis cell division. The meiosis cell division starts with the prophase 1. It goes into metaphase 1, anaphase 1, telophase 1, where the cell divides into two. In meiosis 1, the cell divides into two at telophase stage. Again, these two cells participate and go into the meiosis 2, where at the last stage, that is the telophase 2, the two cells divides into four haploid daughter cells. Meiosis is divided into following phases. Meiosis 1, it is cell division unique to germ Meiosis 1, it is cell division unique to germ cells and divided into interphase 1, where the DNA is copied, prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2, it is similar to mitosis and divided into prophase 2, metaphase 2, anaphase 2, telophase 2. This is the diagram which explains meiosis 1 and meiosis 2. In the next slides, each is explained by magnifying the image. This is the meiosis 1 image, which includes prophase 1, metaphase 1, anaphase 1, and telophase 1 and cytokinesis. Prophase 1 explains the chromosomes condense and the nuclear envelopes break down in prophase 1. Metaphase 1 shows the pairing of homologous chromosomes. Anaphase 1 shows the movement of the chromosomes to the opposite poles of the cells. Telophase 1 shows chromosomes gather at the poles of the cells and the cytoplasm divides. The interface of meiosis 1. The DNA in the cell is copied, resulting in two identical full sets of chromosomes. Outside of the nucleus are two centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. During interphase, microtubules extends from these centrosomes. Meiosis 1, prophase 1. It is the longest phase of the meiosis 1. During this phase, homologous chromosomes condense and pairs. The nuclear envelopes breaks down. Crossing over occurs and DNA segments are exchanged, known as recombination. This is the figure which shows that the homologous chromosomes have condensed and are paired. Nuclear envelope is getting degenerated, that is, it is in the form of fragments, and there is chiasma is shown. Prophase 1 is further divided into 5 stages: leptotene, zygotene, pectine, diplotene, and dikinesis. This is the diagram which explains the prophase 1 stages, which is leptotene, zygotene, pectine, diplotene, and dikinesis. Let us explain in detail each one. Leptotene, it is the first stage of prophase 1. Each individual chromosomes exist with two sister chromatids. Elements of synaptonemal complex assemble. Condensation and coiling of chromosomes occur. This is the picture which shows the individual chromosome exists with two sister chromatid and they are linked with the centromere in the center. Zygotene is the next stage after leptotene. Chromosomes align as homologous pair of chromosomes. Synapses of homologous chromosomes occur. The paired chromosomes are known as bivalent or tetrad. The picture is showing the synapse of the chromosomes. Synapse means the pairing of the homologous chromosomes. Here, the picture is showing the pairing of the chromosomes. Pectine, during this stage, crossing over and homologous recombination occurs. Paired chromosomes shorten and thicken. Chiasmata is formed where homologous chromosomes exchange their segments. The picture here is showing the crossing over of the homologous chromosomes. They are paired and shorten. Next stage is the diplotene stage of prophase 1. Homologous chromosomes start to separate into two pairs of chromatids. The synaptonemal complex disassemble. Chromosomes remain bound at the chiasmata. Chiasmata, the figure is showing the diplotene stage where the chiasma is marked. Chiasma is the point of contact of the two chromosomes. Here, the synaptonemal complex disassemble. The next stage of prophase 1 is the dikinesis. Chromosomes condenses and four parts of the tetrads are visible. The separation of the homologous chromosome is complete and crossing over has occurred. The nucleoli disappears, nuclear membrane disintegrate, mitotic spindle starts to form. This is the figure which shows that there is a formation of the spindle fiber in dikinesis of prophase 1. The next stage after prophase 1, which is the longest phase of meiosis, enters into the metaphase 1. Here, the pairs of homologous chromosomes move to the equator of the cell and are aligned on the metaphasic plate. The replicated chromosomes are joined together via protein known as cohesin. This is the figure which explains that the chromosomes aligned themselves at the equator, that is called as the metaphasic plate. That is called as the metaphase plate. Anaphase 1 of meiosis 1, kinetochore microtubules shorten, pulling homologous chromosomes to the opposite poles of the cell. The cohesin protein complex from the chromosome arms is degraded, while the cohesin surrounding the centromere remains safe. This allows the sister chromatids to attach together while homologs are segregated. This is the figure which shows that the kinetochore is pulling the sister chromatids to the opposite poles. So the pulling of the chromosomes towards the opposite pole is called as the anaphase stage. The last stage of meiosis 1 is the telophase 1. Each daughter cell has half the number of chromosomes as compared to parent cell. The microtubules that forms the spindle starts to disappears. The chromosomes again forms chromatin and gather at the poles of the cells. Sister chromatids remain attached during telophase 1. The cytoplasm divides. This is the figure which shows the cell is dividing into two daughter cells. Meiosis 2. Let us see in detail the meiosis 2. It is the second meiotic division. It is similar to mitosis, but the genetic results are different. It results in the formation of 4 haploid cells from the 2 haploid cells produced in the meiosis 1. The four main steps of meiosis 2 are as follows: prophase 2, metaphase 2, anaphase 2, telophase 2, followed by the cytokinesis. This is the figure which explains the meiosis 2. Prophase 2 shows the spindle formation. Metaphase 2 shows the chromosomes are aligned at the equator. Anaphase 2, where the chromatids move to the opposite poles. Telophase 2 shows a nuclear envelope forms around each set of the chromosomes and the cytoplasm divides. And in telophase 2 of meiosis 2, there are 4 daughter cells which are haploid are formed. So let us go in detail. Prophase 2. In this phase, nucleoli and nuclear membrane disappears. Shortening and thickening of the chromatids occurs during this phase. Centrosomes moves to the poles and spindle fibers are formed for the second meiotic division. This is the figure which shows there is formation of the spindle fibers and the thickening of the chromosome takes place. The next stage, it goes into the metaphase 2. In this phase, the centromeres are present with 2 kinetochores that are attached to the spindle fibers from centrosomes at the opposite poles. The metaphasic plate is rotated by 90 degrees when compared to meiosis 1, perpendicular to the previous plate. The figure explains that the metaphase 2 chromosomes line at the equator. It goes into the next phase, that is the anaphase 2 after the metaphase 2. It is accompanied by the sister chromatid segregation. Centromeres divide. Remain of the cohesin is degraded to allow sister chromatids segregation. The chromatids move to the opposite poles of the cell. The figure here explains that the chromatids move to the opposite poles of the cell. So, this is moving of the chromatids towards the opposite pole of the cell is called as the anaphase, and in meiosis it is anaphase 2. The last stage of meiosis 2 is the telophase 2, which is accompanied by the cytokinesis. Telophase 2 phase is like the telophase 1. Chromosomes move to the opposite poles and begin to decondense. Nuclear envelope reorganizes around each set of chromosomes to create two new cell nuclei. This is the last phase of the meiosis. However, cell division is complete with cytokinesis. This is the figure which shows that there is the formation of the nuclear envelope and it surrounds each set of the chromosomes. Here, the cytoplasm divides and complete the cell division. Cytokinesis means division of the cytoplasm into two cells. Cleavage or cell plate formation starts to produce four daughter cells, each with half a set of chromosomes which are haploid. In males, these four cells are all germ. In males, these four cells are all sperm cells. In females, one of the cell is an egg cell, while the other three are polar bodies, which are small cells that do not develop into eggs. The event of meiosis 2 are analogous to those of mitotic division, although the number of chromosomes involved has been halved. Meiosis generates genetic diversity through the exchange of genetic material between homologous chromosomes during meiosis 1. The random alignment of the maternal paternal chromosomes in meiosis 1. The random alignment of the sister chromatids at meiosis 2. Let us see the significance of the meiosis in plants. Meiosis is very much necessary part of the life cycle of sexually reproducing animals and plants, as it helps in restoring the definite number of chromosomes, the characteristic of a species. The crossing over of genes between homologous chromosomes helps in exchange of genes, leading to the formation of new recombinants. Meiosis is most essential for the completion of life cycle of a plant, as it brings a change from diploid to haploid generation. These are the references. Thank you, students.